Good afternoon, everyone. My name is Wei, doing PhD in UCL. Today I'm going to present you my PhD project. The topic is Artificial Hypoxia-Inducible Factor Stabilization for Enhanced Cell and Tissue Construct Survival, supervised by Professor Barry Fuller and Dr. Gavin Zhao. First of all, why is it important? Stem cell therapy we all know stem cell therapy is a promising clinical therapy in regenerative medicines. However, there's two obstacles to the approach. In vitro study, cell expansions, storage, and transportations requires improved cell survival. During cryopreservation, cold hypoxia and the use of DMSO will decrease the amount of injectable cells and also affect the stabilities of the proliferative and phenotype. Even the amount of cells is achieved for implantations. Large of a number of cells still dies in the damaged area due to deviations of nutrient and oxygens. And hypoxia preconditioning is preconditioning the cells in no oxygen conditions, and which can enhance the transcription of hypoxia-responsive elements to allow cellular adaptations to adverse conditions. And recent study has shown that preconditioning cells can enhance cell functionalities. For example, liver regeneration, they shows increased albumin levels, and other study also shows increased androgenetic factors as well. In vivo study also shows quite similar results as in vitro, such as increasing the VEGF and also anti-apoptosis marker. And the most important factors in this pathway is hypoxia-inducible factor 1 alpha, HIF1 alpha. Is a transcription factor responsible for cellular oxygen sensing in order to promote cell survival. In normoxia conditions, HIF1 alpha is hyoxygenized by an enzyme called PHD enzyme, which is activated by oxygens and target the HIF1 alpha for the degradation pathway. But in normoxia conditions, low oxygen conditions, PHD will be inhibited and HIF1 alpha will be stabilized in cytoplasm, and ultimately will translocate to the nucleus and bind to the HIF1 alpha, HIF1 betas, and to transcript those post-survival genes. And these genes can enhance cell survival. For examples, GLT1, for example, glucose transporters, GLUT13, which balance the energy supplies and demands. And also, for example, VEGF stimulate new blood vessel formations. And the other example is BCL21, which prevent the hypoxia-induced cell death. And the aim of my experiment is using hypoxia-mimetic agents, such as cobalt and DFOs, to activate this pathway in normal oxygen conditions, rather than hypoxia preconditioning, because low oxygen conditions mostly will inhibit cell growth and increase ischemic injury. And using the chemical, one of the advantage is we can use scaffold to control the release system, and which can induce the oxygen. We can induce this pathway in the normal oxygen condition as well. So the aim of my study is to determine if hypoxia mimetic agents can enhance cell survival and proliferations in 4° and -80° preservations. And the objective of my study is to develop cold and freezing preservations model to show hypoxia preconditioning cells can enhance cell survival in vitro. And following, I want to demonstrate hypoxia mimetic agents. Preconditioned cells can induce similar effects as hypoxia preconditioning. Most research groups, they using 24 hours. They've been preconditioning the cells for 24 hours at 1% of oxygen conditions. Therefore, in my experiment, I'm going to preconditioning the cells in 1% oxygen concentrations for 24 hours. So in this slide, I'm showing the optimizations of the DMSO. We can see in here, in this graph, it's showing the dead and live cells by using different concentration of DMSOs, and we can see that reducing the concentration of DMSO also decreasing the number of live cells, as the blue bar shows. And that same decreasing trends can be observed in the metabolic activities and the total DNA assays. So for my initial study, I'm going to use 0% DMSO to cryopreserve the cells, and I hope I can see a great opportunity using 0% DMSO, because I think there will be a great opportunity to compare the samples with the controls, 10% DMSO. Here is the procedure. I'm working in my physics study, and I'm using fibroblasts from human dermal fibroblasts from elders to precondition the cells in normoxia and hypoxia incubators with or without cobalt for 24 hours. And after 24 hours, the cell will freezing down in FBS alone for another 24 hours. And later, I will seed in the cell in 96 well plates to see the recovery and survival. Here's the data show the metabolic activities on the left-hand side, and total proliferation rates on the left-hand side. And we can see the HP stand for hypoxia preconditioning, the cell preconditioning in the hypoxia. There's a significant difference in the metabolic activities compared the rest. But however, in the proliferation rate, we can see that cobalt cell preconditioning in cobalt in normoxia condition have similar proliferation rates as the hypoxia preconditioning cells. So in here, it can say cobalt have similar proliferation rates as hypoxia preconditioning cells at day 7. And here is the other study on preconditionings, on preservations at 4 degrees. And HepG2 cells I'm going to use in here, and the cells incubate for normoxia conditions for 24 hours for maximum cell attachment, and later the cell will will freezing down the whole place. The media will be removed, and then put the perfusion liquid with different hypoxemic agents, and freezing down at 4 degree fridge for different length of times during the course of the experiment. And the other one is same maximum the cell attachment on the 96 well plates at normal Mossier incubators, and later translocate to the hypoxia incubator for preconditioning. And after 24 hours hypoxia preconditioning, the cell will, and the media will be removed and will replaced by the hypoxia perfusion liquid without any hypoxemic agents for the same time length, 30 minutes, 4 hours, 12 hours, and 24 hours in the fridge. And later sitting them in, remove the, and later after this time length, and the perfusion liquid will be removed and replaced by media and put it back to the Mossier conditions. And the categorization, I'm working to categorize the metabolactivity. I'm using alamabut and cell proliferations, total DNA assay kit, and cytotoxicity. We'll test on the cell tox screen cytotoxicities. And the ROS, we're using the dye to see the attachment, to see the ROS. So in here, the cold with storage preservation affects cell survival. And we can see that is the control, the cell without any cold preservation. We can see increasing the amount, increasing the time of cold preservation, we reducing the amount of cell as well, and also reduce the metabolic activities. So the duration of cold storage preservation could affect cold survival in here. And thus, hypoxia preconditioning can increase cell survival. After the previous step, I freezing down the cells for 3 minutes, and then 4 hours, 12 hours, and 24 hours. The preconditioning cells seems have higher survival regarding the metabolic activities and the total DNA. But we cannot. There's no any signs of cell survival at 24 hours at 24 hours preservations, cold preservations, compared to the controls. That's even hypoxia preconditioning cells have higher cell survival regarding the metabolic activities and total DNAs, but still no survival on the 24 hours cryopreservations. So in here, I want to see cell recovery during on day 3 and day 7, and the same trends as the survival is seen on preconditioning cells, but still no signs of 24 hours 24-hour preservations, co-preservations. So we can see that hypoxia preconditioning can increase cell recovery and survival during the course of the experiment, except the long-term cryopreservation, long-term co-preservations. Does hypoxia mimetic agents have similar effect as hypoxia preconditioning on cell survival? The answer is on the survival is no. On the survival, the cell seems not react that well as hypoxia preconditioning cells. But however, we can see there's a little amount of DNA after 24-hour cold preservations with different amounts of hypoxemic agent, the cobalt and the DMOB. But we cannot see. But however, there's no any metabolic activities on the first state. So let's go to the next slide. It shows more promising for the hypoxia mimetic agents. Particularly on day 3, we can see that even the hypoxia preconditioning cells did not show any metabolic activities, and even the DNA amount we hardly to see, but we can see the cells preconditioning with the hypoxia mimic agents, the cobalt and the MOT, have higher cell number and also higher cell metabolic activities. So, hypoxia mimic agents demonstrate better cell survival at 24-hour cold storage preservation at day 3 and day 7. And to see the cytotoxicity, we can see generally this range of cobalt concentration and the MOC concentrations didn't have higher amounts of cell death compared to the control. So hypoxia, so that is a promising result to use among this range of concentration as well. And the other interesting data I would like to show is like ROS, the hypoxia mimic agent, to increase the ROS, reactive oxygen species, within the 2 hours after you bring up from the freezing. So the data may suggest that this induced ROS regeneration can enhance the cell survival for the long-term cold storage preservations. And in my conclusions, freezing and cold storage preservation shows hypoxia preconditioning and hypoxia mimic agents can enhance cell recovery and survival during the course of experiment. Hypoxia mimic agents has lower cytotoxicity, and the data suggests that hypoxia mimic agent may induce ROS production to enhance cell survival. And also, some research have already demonstrated maybe they are also interact with the cellular metabolic activities for the adaptations to the adverse conditions. Thank you.